Hey guys, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 10, Controlling Microbial Growth in the Body. So basically chemotherapy, antimicrobial chemotherapy, which is drugs. We've been working our way through how these drugs work. What is their mechanism of action? We said everything has to be evaluated in regards to selective toxicity. Selectively toxic to the pathogen without harming the host. So we've worked through cell walls, protein synthesis, cell membranes, and metabolic pathways. And the last category is nucleic acids. This category, pathogens have nucleic acids, we have nucleic acids, and they're basically the same thing, a lot of similarities from that. So this is kind of a poor, be careful category. We can get around this some ways. But we do have some drugs in this category, but it's definitely a be careful kind of deal. So let's look at our drugs in this category of inhibiting nucleic acid synthesis. Okay, so inhibiting synthesis, we can block DNA replication. We can block mRNA transcription, but again, this is very similar to host and pathogen. So these can often affect both eukaryotic and prokaryotic cells. So we have to watch for that. Therefore, drugs that just block replication and just block transcription, not usually used. We can block some enzymes, and we can use some nucleotide analogs. So let's look at both of those options. First, what I want to talk about is disrupting an enzyme. So even though replication and transcription are very similar in prokaryotes and eukaryotes, there are a few differences. If we target those differences, we can achieve selective toxicity. So we're going to disrupt enzymes used in nucleic acid synthesis in prokaryotes, not the ones that are used in eukaryotes. So drugs in these categories, the ones in purple that I want you to know as an example are quinolones and fluoroquinolones. They act against an enzyme called DNA gyrase. You have heard this enzyme in a previous chapter called topoisomerase. If you remember back to DNA replication, we talked about that initiation of replication, and you had helicase. And helicase unzipped the DNA, and it moved through and it broke those hydrogen bonds. And then you had single-stranded binding proteins that came in. And then you had an enzyme called topoisomerase, and that kept it from being supercoiled. As the two strands of nucleic acid were being separated, we didn't want the others to get all supercoiled. Sorry, I don't have my visuals on that one. But topoisomerase is the enzyme that does that. Another name for topoisomerase is gyrase. So gyrase is the enzyme that protects that supercoiling of DNA, and there is a drug that acts on gyrase. This is an enzyme, again, that's only on bacterial DNA replication. Eukaryotes do it a little differently. So this is an example of how selective toxicity can be achieved. We're going to come back to reverse transcriptase in a minute. What's another way we can do? Another thing we can use in this category are drugs that are actually nucleic acid analogs. So down here, we've got some nucleosides, our sugar and our bases. We have adenosine, guanosine, cytidine, and thymidine. So those are the names of the nucleosides from that. This is what they're supposed to be. This is what eukaryotes have. This is what prokaryotes have. This is the normal, but there are molecules that are similar but not the same. So here is thymidine here. There is a molecule called azethymidine that's very similar. If you want to look, there's an amino group here instead of a hydroxyl group, but otherwise they are similar. So an analog is a molecule that looks like something, but it's not quite the same. So this is a chemical, a molecule that is similar but not identical to thymidine. All right, if I have a bacteria cell that's undergoing replication and I add a whole bunch of this AZT, this nucleic acid analog, azethymidine, what happens is bacteria are replicating very quickly, and they are grabbing those thymines and they're grabbing those very quickly and incorporating them into nucleic acid. When you are doing something quickly, you are likely to grab the wrong thing. You're likely to make a mistake. So if I've got this molecule in this environment, this drug, this azetothymidine that looks similar, but it's not the same thing, that bacteria cell is going to grab it and incorporate it into the nucleic acids by accident. When it does that, it's not the same, it's not identical, so that nucleic acid is going to be misshapen and it's not going to bind correctly. So I'm adding in mutations. So the more AZT I have in there, the more it gets incorporated into these very quickly replicating bacteria and viruses, the more mistakes, mutations that are going to be made, and the more likely that that virus or bacteria is not going to be able to replicate and it's going to die. So that's how nucleic acid analogs work. All right, so they get substituted by these enzymes. The result in distorted nucleic acids that don't get replicated and they die. These drugs are especially effective against viruses because viruses replicate quickly. Some bacteria. The other thing that they're good about is cancer drugs. Some of our cancer drugs are nucleic acid analogs. And the tumor cells are also replicating quickly. They will grab these analogs and get incorporated into them instead, and the cancer, the tumor cells will have a whole bunch of mutations, and then the tumor will die. So that works. AZT, the one that I used in an example, this is a drug that is part of the drug cocktail that we use to treat HIV infection. So HIV can be treated by this AZT. It was the very first drug that we had to treat HIV. HIV is a virus. It's replicating very quickly. It incorporates more of this AZT than our normal cells do, and we can get rid of the virus that way, or it's effective against that. The other thing we can use against HIV is we can target an enzyme that's only found in the virus and not found in the host. If we remember back to our virus chapter, we talked about retroviruses. Retroviruses come in as positive single-stranded RNA, but then they get converted into DNA. And that's a bad thing because they get to hide in the host nucleic acid. The enzyme that the virus makes, or that has the DNA, the instructions to make, it makes the host cell make it, is called reverse transcriptase. And that allows it to go from RNA to DNA. This is an enzyme that we don't need. We never have a need to go from RNA to DNA for the host. So if we can target this reverse transcriptase, we can have an antiviral drug, a drug that's going to stop the virus without harming the host. So that's a way that we can target differences in DNA replication between a virus and a host. All right, so nucleic acid analogs and those. All right, antiviral drugs, just one more note on that. We kind of drew our circles. It is very hard to get selective toxicity with viruses. We do not have a lot of antiviral drugs. They're very difficult. So SARS-CoV-2 is a virus. It's very difficult to come up with drugs for that. The drugs we do have work by stopping penetration or attachment. Again, if we can get rid of the SARS-CoV-2 envelope, so that's what you're spraying bleach on your kitchen counters, if we can stop the envelope, that's good. If we can have a drug that will do that. But you remember, an envelope is just part of a host cell membrane. It's very hard to have drugs that interfere with host cell membranes. But if we can target some of those glycoproteins, those spikes, maybe that would work. If we can block transcription or translation of the virus without viral proteins without stopping the host, or if we can prevent them from putting the virions together. But these are tough. And the other thing you need to remember about antiviral drugs is antiviral drugs protect uninfected cells, but once the virus has gotten into that cell, if you remember back to chapter 13, once that virus gets in, that cell is going to be a goner. There may be some things it can do to slow it down or protect it or protect other cells, but that one's a goner. Same thing with the drugs. The antiviral drugs we have protect uninfected cells. So one example of an antiviral drug we have is aciclovir? Abreva. Abreva is one of the common names for it, but you get those cold sores. Remember we talked about the herpes simplex viruses, and you get those cold sores, and it affects epithelial cells. We talked about that when we talked about latency in the virus. It affects the epithelial cells, causes damage and kills epithelial cells. You get that blister. Meanwhile, the virus then moves on and integrates or infects neural cells, but this time it integrates in there and it stays in those neural cells. Our epithelial cells repair, our interferon, our immune system gets it, kicks the virus out, our sores repair, but we still have that provirus hiding in the neural cells. So something triggers it, stress, hormones, something triggers it, comes out of the neural cell, excises from the DNA, comes back into epithelial cells, and you get another breakout. There are some drugs that we can use to make that breakout less severe. So when you first start feeling that tingling that coming on, if you've had that before, there's a tingle or something that you feel from doing that. Sometimes you can't even see that you're having damage, but you know, you can feel it. Start using those antivirals, and that will protect the uninfected cells. You may still get a breakout, but it will be less cells involved, so less damage. So that's how the antivirals work. Remember that they will only protect uninfected cells. And then your immune system's going to have to take care of the virus. Okay, antiviral drugs, I think we are going to stop there, and we will do the next the next series of videos. We've been doing a whole series on mechanisms of action, and now we're going to talk about some things we have to consider when we're doing clinical applications. So this is kind of a fun section too. Okay, stay with me. Don't get bogged down in those mechanisms of actions. They're very important, but I know some of you guys that are tired of the cell biology, this is kind of back to that. So understand the concepts and then keep going forward in this chapter. Okay, thanks.